Before you get started, ensure you have the protein and all the components of Biacore CapTag capture kit you need for protein kinetic analysis. You will also need Amersham microspin G50 columns, Series S sensor chip cap, Biacore regeneration kit cap, and the protein. We are going to use Biacore 1S Plus for the kinetic analysis. Begin your analysis by rehydrating Series S sensor chip cap. This can be done either by docking the sensor chip in the instrument and leaving on standby with running buffer overnight, or if you don't have access to a Biacore SPR system the night before your experiment, you can rehydrate the sensor chip by placing it in a tube of water or HBS EP Plus buffer overnight. While the sensor chip is rehydrating, perform the Biacore CapTag conjugation by following the instructions for use. Note that this procedure can also be done in advance, provided the protein remains stable during storage. This will be protein dependent. The first step is to conjugate the protein with the CapTag. Perform buffer exchange of 50 microliter of ligand protein using Amersham microspin G50 column equilibrated in Biacore conjugation buffer. Add the required volume of Biacore ligand activator based on your protein size. This information can be found in the instructions for use. Mix and incubate for 1 hour, protected from light. Purify the activated protein using a microspin G50 column. And then add the recommended volume of Biacore CapTag as recommended in the instructions for use. Mix and incubate for another hour. To complete the conjugation procedure, use microspin G50 column to remove unconjugated Biacore CapTag, leaving you with Biacore CapTag ligand conjugate. Performing these protein tagging steps outside of the instrument allows you to run multiple projects simultaneously. Your ligand conjugate is now ready for protein interaction analysis using Series S sensor chip cap. Return to your Biacore instrument and ensure Series S sensor chip cap is already hydrated and docked. Open Biacore Insight Control software and open the method Capture Level Scouting using Biacore CapTag Capture Kit. If this method is not already available, it can be downloaded from the Biacore software downloads page. Enter your sample name. If required, edit dilution factor and contact time. Prepare the dilution series of the ligand conjugate in running buffer and pipette the samples into vials or plates. Load the samples onto the instrument. To open the door to the sample hotel, press the button on the hotel door. Pull the lever with the arrow sign on the sample tray toward you to open it. Place your reagent rack on the sample tray in the sample hotel. The microplate slot is to the left, and the reagent rack slot is to the right. Carefully push the reagent rack all the way in until you hear a clicking sound. The lever automatically closes around the reagent rack. Close the hotel door when you are done. Now start the run. Once the run finishes, open the Capture Level Scouting run and apply the predefined evaluation method, Capture Level Scouting, using Biacore CapTag Capture Kit evaluation method, also downloadable from the Biacore software downloads page. Examine the QC sensorgrams and QC plots to check the run has proceeded as normal. Check the plot of capture level versus dilution factor and select the dilution that results in the desired capture level. The desired capture level can be calculated using our online tool, SPR Sensor Surface Density Calculator, which can be found on cytiva.com/Biacore Tools or by referring to Biacore CapTag Capture Kit instructions for use. Next, open the run method Single Cycle Kinetics using Biacore CapTag Capture, also downloadable from our website. Enter solution names for the 3 solutions, running buffer, ligand conjugate, and analyte. Enter the ligand conjugate dilution factor. Next, either use the predefined values or edit the analyte concentration range, contact time, and dissociation time according to what is known about the interaction. Dilute the ligand conjugate in running buffer and prepare a dilution series of the analyte in running buffer. Pipette the ligand conjugate, analyte dilution series, and regeneration solutions into vials or plates, as stated in the control software. Load the samples in your Biacore instrument and click Run. Once the run is finished, open the kinetics run and apply the predefined evaluation method, single-cycle kinetics using capture. Examine the sensorgrams and plot items. Examine the kinetic items, including the blank sensorgram, residuals plot, kinetic parameters, and fit quality. We recommend using Biacore Simulate to confirm your kinetic parameters make sense. This can also be found on cytiva.com/Biacore tools. Enter the kinetic parameters from the evaluation into Biacore Simulate and assess if the sensorgrams look similar to those you have obtained experimentally. Depending on how much is known about the interaction, you may have chosen an analyte concentration that is too high, too low, or needs shorter or longer contact times. If this is the case, you can use Biacore Simulate to figure out ideal parameters. Since Series S sensor chip cap can be reused, you could now proceed directly to repeat the experiment with optimized conditions using the same sensor chip. If you are happy with your data, you can now either leave the sensor chip docked in the Biacore instrument ready for the next set of experiments, or undock it. In Biacore Insight Control software, go to Instrument Control and select Change Chip. Click Undock Chip. This will undock the sensor chip currently in the instrument. Now remove the sensor chip from the protective cassette with a pair of tweezers and place in a 50 milliliter tube containing HBS buffer. Cap the tube securely and store at 2 to 8 degrees Celsius. The sensor chip can be stored for up to 2 months and withstand at least 150 cycles. You can now start a new run or allow other scientists to perform their experiments. To learn more, visit cytiva.com/Biacore CapTag.